Thank you. It's very nice to be here and meet you again, everyone. So yes, as Brian said, I'm representing the industries today. I'm Elisa Napoli. I actually worked in metrology before at NIST during my PhD. And I'm really fond of metrology. So I continue this work also in Bayer because we need that, especially when we have multiple nuclides to work with. A bit about Bayer. Bayer is a global enterprise located in all around the world, but I come from Norway, which is called Bayer AS, which is a company that acquired the old Algeta that produce Sofigo, which you have seen in the previous talk. Is a company that work on different fields. My field is the pharmaceuticals, and we focus on oncology. The organization has over 150 employees in Norway and from many nationalities, and we work on different fields, and the organization have also different backgrounds. I'm a physicist, and it's very important to have people with many different backgrounds. So to show you a bit the journey of our nuclides. We started with Sofigo, as I said, in 2014, when it was approved by the FDA. And then we moved to different nuclides, thorium-227 and now actinium-225. And we are still progressing on different novel targeting. As you know, it's important not only to have a radionuclide that you can use for therapy, but also you have to image. So we also are involved with that and do a lot of preclinical trials and now clinical trials as well. And now I wanted to show you a short video about how we actually prepare our products and how does it work. So enjoy.
Hi, I'm Ingrid, and I work as a cancer researcher at Bayer. Cancer, unfortunately, affects most of us in one way or another during our lifetime. But the good news is that researchers worldwide are working hard to provide better treatment options to the patients. Today, I'm here to share how we at Bayer Oslo contribute to the evolution of novel cancer therapies. When a person is diagnosed with cancer, most of the patient's body is still populated by the healthy cells. Let's call them the good guys. The sneaky cancer cells, or bad guys, hide within the group of good guys, either in clusters, known as tumors, or as individual cells. My team and I are waging a war against cancer. But war, as we all know, has an ugly face too. It's not just the bad guys who get hit, but sometimes the good guys get equally hurt. For example, nowadays the primary method of removing a cluster of bad guys is through surgery assisted with therapies such as chemotherapy and radiotherapy. However, these traditional approaches have two main drawbacks. They fail to differentiate between the good and bad guys, and there's always a risk that traces of bad guys are left behind after treatment. Here in Oslo, we are developing a new type of cancer therapy which leverages radiation in a new way. Now, I know that most people often associate radiation among the factors that can actually cause cancer, but our experience shows that with the right knowledge and expertise, we can actually use it to treat cancer. Let me explain 3 different ways in which radiation can be used to treat cancer with the help of an analogy. Consider a stable atom as a happy person. For them, all is well, and I do not want anything to change. A radioactive atom, on the other hand, would be like an angry and frustrated person who, in order to make up and achieve stability, needs to release a lot of energy. This energy is what we know as radiation. One way this energy is released is in waves. Imagine this person screaming loudly. The piercing scream can penetrate through thick walls and damage nearby objects. These radioactive waves represent the first generation radiation treatment and are known as gamma radiation. These waves can be directed to the area where the cancer is located. They can penetrate the body tissue and even reach tumors deep within the body. But these waves fail to differentiate between the good guys and the bad ones, hence often harming the good guys and causing damage to helpless tissue. Release of energy can also happen in the form of particles. There are in general two types. First, consider the angry and frustrated person throwing something, for instance, a coffee mug. This particle can do larger damage than the piercing scream, but at a comparatively shorter distance. This is called beta radiation and is being studied in an emerging field within cancer therapies. The second particle type is what we at Bayer work with daily in our lab. It's called alpha radiation. This is a large and heavy particle that can do a lot of damage, but within a very short distance. Imagine the angry person wearing boxing gloves and punching really hard. It's a heavy punch that can do immense harm, but the traveling distance is limited. These alpha particle boxing gloves must get close enough to the place where the bad guys are hiding. If they are placed outside the human body, they won't even be able to penetrate through the skin. We believe we have a way to address this challenge. Years of research have helped us identify the distinct traits of the good versus bad guys and design special drugs that can bind to the bad ones. These drugs, combined with the alpha radiation particles, act as precise warheads that lead the damaging radioactivity specifically to the bad guys, bind to them, and destroy them. The good news is that due to the limited traveling distance of the alpha particle, we prevent the good guys from getting hurt. This is called targeted cancer therapy and constitutes the next generation of cancer treatment. To summarize, different properties of radiation affect the human body differently. And by using this knowledge, we can design drugs that can successfully target and hit the cancer cells without hurting the normal tissue. I am proud to be part of a team that's working relentlessly to develop new and sophisticated cancer therapies. I believe that every day we are one step closer to winning our war against cancer. But we have lots of work to do, so excuse me, I must hurry back to my team.
I hope you liked the video and that maybe answers some questions from before as well. So to go back to Bayer, we have a very nice lab that has a very nice view over the sea. Some of you know, have been there. And we are equipped with all most important GMP analysis techniques for radionuclides. Among our detectors, we have different kind of detectors that can help us measure from the small impurity in the alpha emitting impurities to beta impurities through the gamma field. And also we do mass spectrometry analysis. And today I'm going to talk a bit about the dose calibrators concerning dial setting determination, because it's probably most relevant for the metrology part. I work in quality control, and when I say that I work in quality control, people think this. is not really this, is more focused on development and validation of analytical methods, GMP analytical methods to assess the quality of radiopharmaceuticals. Therefore, when it comes to radionuclide, we look at the radionuclidic impurities profile and make sure that everything is good. And then we can apply for to the FDA or to EMA for clinical trials applications. I also work with the standardization and geometry studies, which are really important. And this is one of the challenges that we face, since this is also the question from Brian before. The main challenges that we face is that we have many different geometries and therefore also many different sites. So we have to make sure that everyone is traceable to one common standard. And of course, NIST or NPL or the others can help with that, and maybe calibrator ionization chambers also for specific geometries, or make sure that we can have everything smoother. And also we work on other tasks, but let's see an example. For instance, for Sofigo, we are traceable to NIST, all our clinical sites and all our contract manufacturing organization and ourselves, we are traceable to NIST. And what we do is that we prepare an ampule in the NIST geometry that they send us. We fill it with the volume that they requested. We flame seal it and we ship it to NIST. And then NIST measures it in their ionization chamber. And then send this over to other contract manufacturing organization. And we make sure that everyone is traceable to the same source. And after a while, then we receive a certificate, and the certificate reports the activity of the source. Then we do this check yearly to make sure that the dial setting that everyone is using doesn't have any considerable bias concerning the difference between what they say and what NIST says. We do the same with actinium-225. I think we are the only one so far that has made this traceability chain. We prepare standards in-house, and we make sure that PTB, the German Metrology Institution, receive a source from us that they will measure, and we make sure that we prepare twin vials of known mass, and we send those to our sites. Then we receive a certificate from PTB, and we trace back on the activity of each vial, and therefore calibrate new dose calibrators or check those style settings. Yes, and that was all for me.